Yes. So what have they said? We are talking about living cells. All the living cells are quite close. And what is the thickest part of the tree we see is the stem. And in the stem also, all the living cells are in the external part. So the distance for diffusion is very low. That is what is the third reason. Yes. And there is the other point is there is the openings are lenticels and the interior is dead. Mechanical support, we've already seen this, so we are moving on. Okay. This is also facilitated by the loose packing of parenchyma cells. This is also important. What is also facilitated? The oxygen exchange, the gaseous exchange is also facilitated by loose packing of parenchyma cells because in the air, they provide a network of air spaces. Okay, they provide a interconnected network of air spaces. The complete combustion of glucose, which produces carbon dioxide and water and as end products, yields energy, most of it is given out as heat. If this energy is to be useful to the cell, it should be able to utilize it to synthesize other molecules that the cell requires. The strategy that the plant uses is to catabolize the glucose molecule. Catabolize means break down the glucose molecule in such a way that not all of them are liberated as heat. The key is to oxidize glucose not in one step, but in several small steps, enabling some steps to just be large enough so that the energy released can be coupled to ATP synthesis. That is, the energy will be just enough for synthesizing an ATP. How this is done essentially is the story of respiration, which we're going to study now. During the process of respiration, oxygen is utilized, and carbon dioxide, water, energy are released as products. The combustion requires oxygen, but some cells live where oxygen may or may not be available. So what happens to those organisms where oxygen is not available? We'll see about that. Anaerobic respiration. There are sufficient reasons to believe that the first cells on the planet that lacked oxygen, we'll see about this in evolution in the next year. Even among present day living organisms, we know several organisms which are adapted to anaerobic conditions. Some of these organisms are facultative anaerobic, that is, they can live only, they do all the stuff only without oxygen. They have to be anaerobic. That is what is obligate anaerobe. So some are obligate anaerobe. In any case, all living organisms retain the enzymatic machinery. They retain the enzymatic machinery to partially oxidize glucose without the help of oxygen. That is whether they are facultative anaerobe, whether they are obligate anaerobe, or whether they are aerobe. Everything there is one common pathway. What is that pathway? Glucose to pyruvic acid, the commonly known, the famous pathway, the glycolysis pathway. We'll be studying glycolysis. So the term glycolysis has originated from Greek words glycos means sugar, lysis means splitting. The theme of glycolysis was given by Gustav Embeden, Otto Merhoff, and Zay Parnas. Now you have a doubt, you would be asking like you can have a doubt like should we memorize this? Is this essential? Just give it a look. I've now not seen this much in questions, but it's okay to give a look. Gustav Embeden, Otto Merhoff, and Parnas. Like they also call it the EMP pathway. E for Embeden, M for Merhoff, and P for Parnas. And is often referred to as the EMP pathway. Okay, it's in the next line, sorry. In anaerobic organisms, it is the only process in respiration. Glycolysis occurs in cytoplasm of the cell and is present in 1 minute. Yeah, and is present in all living organisms. In this process, glucose undergoes partial oxidation. What do they say before? What is the first statement? Glycolysis occurs in cytoplasm of the cell. So you should glycolysis only like in the total respiration only glycolysis takes place in cytoplasm. All the other stuff after glycolysis itself all happens in the mitochondrial area only. Okay. Yes. What does it form? One glucose forms two molecules of pyruvic acid. Pyruvic acid is a three-carbon compound. In plants, this glucose is derived from sucrose because sucrose is the transporting thing, which is the end product of photosynthesis too, and it's also the storage thing. So this is you should remember. End product of photosynthesis is sucrose. Very important. Or from storage carbohydrates. So what happens? Sucrose is converted into glucose and fructase. What is the enzyme? Invertase. Very simple. And these two monosaccharides readily enter the glycolytic pathway. Glucose and fructose are phosphorylated to give glucose 6-phosphate by the activity of enzyme hexokinase. These enzyme names are there now. These are very important. I'll name the enzyme names in red color. Hexokinase. Okay, this is too much dark. still dark, but it's okay. Hexokinase. This phosphorylated form of glucose then isomerizes to become fructose 6-phosphate. So there are two pathways. What happens? First, there is glucose and there is fructose. Both of them under hexokinase only. What do they form? Glucose forms glucose 6-phosphate and this forms fructose 6-phosphate. And after that it's all common from that. You have to remember where ATP is utilized. ATP is utilized when ATP is used now. From triphosphate it becomes diphosphate. So that phosphate is going somewhere now, and this phosphate is going into glucose to form glucose 6-phosphate. And the second time from it is going to form glucose biphosphate. Two phosphates are there. Okay. This phosphorylated form of glucose then isomerizes to produce fructose 6-phosphate. Subsequent steps of metabolism and fructose and glucose are same. The various steps of glycolysis are depicted in figure 14.1. This is the figure we just saw that. In glycolysis, a set of 10 reactions under the control of different enzymes takes place to produce pyruvic acid. Pyruvic acid without the COH is there now, that is acid. Without the H is pyruvate from glucose. While studying the steps of glycolysis, please note that the steps at which utilization takes place or NADH utilization or synthesis of in ATP or NADH takes place. Okay. ATP is utilized in two places, which we saw for glucose to glucose 6-phosphate and fructose to fructose phosphate to fructose biphosphate. And remember where these phosphates are, in which positions these are there. So first is in 6 and the next is in only 1,6. Okay. This is also important. This people forget. People overlook this fact. So first it is 1, the next one is 1, first it is 6 and after that it is 1. So 1,6 biphosphate. So after this, what happens? Fructose 1,6 bisphosphate is split into dihydroxyacetone phosphate and 3-phosphoglyceraldehyde. Two molecules are there, and there are total, and in the primary thing there are two phosphates, so each of these gets a phosphate, triose phosphate and dihydroxyacetone phosphate. Not triose phosphate, my bad. This is not triose phosphate, this is glyceraldehyde 3-phosphate. Both are triose phosphates. What are triose phosphate? There are 3 carbons. There is O means there is a double bond, and there is a phosphate molecule. These are like, you can say, isomers. So glyceraldehyde. Aldehyde, if it's an aldehyde group, it's glyceraldehyde. If it's an acetone group, it's dihydroxyacetone phosphate. Like there is dihydroxy, and there is acetone, and there is a phosphate. Here, where is the phosphate? It is in third carbon. That is, first carbon is double bond O, second carbon is OH, third carbon is OH, and it has the phosphate molecule here. And here, since carbon cannot have phosphate here, so it's either in this place or this place. Okay, triose phosphates is done. It has been split. Everything is very smooth until now. The next step after splitting, what happens is NADH is going to be synthesized. How are we going to synthesize this NADH? What are we going to transform it into? Before that, you have to remember this very important fact. What is that important fact you have to remember? You have to remember the the letters. 3-phosphoglyceraldehyde is called PGAL. Glyceraldehyde. You shouldn't get confused with this. Very important. Dihydroxyacetone phosphate and 3-phosphoglyceraldehyde. 3-phosphoglyceraldehyde, glyceraldehyde, 3-phosphate, both are same. So don't get confused. They are just shifting the order in which they are saying this. We find that there is one step where NADH is formed. This is when 3-phosphoglyceraldehyde, PGAL, is converted into 1,3-biphosphoglycerate. Okay. So what happens here? NADH is synthesized. But along with that, a phosphate, inorganic phosphate molecule is added. They have not given this in NCERT. See, but it becomes bisphosphate. And there are 2 molecules. That means there are 4 phosphates. Initially, there were only 2 phosphates in one main thing. This suggests that there is an also inorganic phosphorus being added in this process. Okay. And we have got bisphosphate, bisphosphoglyceric acid. 1,3-bisphosphoglyceric acid, BPGA, bisphosphoglycerate or bisphosphoglyceric acid is formed. 2 redox equivalents are removed from PGAL, 2 NADH is formed. That is what they are saying. 2 NADH is formed. Why? There are two molecules of triose phosphates. So two are formed. PGAL is oxidized with inorganic phosphate. PGAL is for to get PB. Okay, BPGA. Okay, that given in NCRT. My bad. The conversion of BPB by bisphosphoglyceric acid to 3-phosphoglyceric acid is also an energy yielding process. So there are two consecutive energy yielding processes. First, we get NADH, and then we are getting ATP. This bisphosphate, one phosphor we are removing, so we just get triose phosphate. Just like the earlier one. Triose phosphate, here also triose phosphate. What is the difference? There, glyceraldehyde, 3-phosphate, here phosphoglyceric acid. Not much of a difference if you ask me. Phosphoglyceric acid and phosphoglyceraldehyde are different. Why are they different? I'm very sorry. Here there is an aldehyde group and here this is an acid group. This is the difference. Although both are triose phosphates, here glyceraldehyde 3-phosphate, here glyceric acid 3-phosphate. Okay, this is something you should note. This is acid group and this one, where is this one? This one is aldehyde group. This is a follow along video, so please mark it in your NCRT. Please underline whatever the important points I'm telling you. Thank you. The conversion of BPPGA into 3-phosphoglyceric acid is also an energy yielding process. This energy is trapped for forming ATP, another ATP synthesized. When is the next ATP synthesized? During the conversion of PEP, phosphoenol pyruvate into pyruvic acid. So after that, they have not told anything in statements. So we will go to the graph and again study the graph. So okay, until triose phosphate, everyone is clear. So what are we doing? We are taking another step. Nothing is here. We will see whatever these steps are lacking, whatever the enzymes are lacking at the end of the video or in a separate video. Here 2-triose phosphate is becoming phosphoglycerate. Not a big change. 2-phosphoglycerate. Here 3-phospho. That is just isomerizing. What is happening here? Position isomerization. If you remember these steps, you don't have to worry about the other stuff. You have to understand. You shouldn't mug up everything. So 3-phosphoglyceric acid, but you have to mug up. You should some stuff. You can't ask me why this 3 is becoming 2. You can't ask me that because we can't answer this. This is how nature is. 3-phosphoglyceric acid and 2-phosphoglycerate. Okay. After this isomerization, after this phosphor shifting has taken place, what happens? We are converting into phosphoenol pyruvate. What is an enol group? That is, if there is a double bond, there is C double bond and another C, this converts into an OH, all, and an ene, an alkene group. This is called enol group. So phosphoglycerate is enolized by along with the release of water. Here it is an acid group now, and from this acid group of water is removed, and after that only phosphoenol pyruvate is formed. This is a very important question. They have asked this several times. Which is a dehydrating step of glycolysis? Phosphoglycerate to phosphoenol pyruvate. And what is the last step of glycolysis? Phosphoenol pyruvate into pyruvic acid with a release of ADP. Here there is phosphoenol pyruvate. Here we are removing this phosphorus to form pyruvic acid. When I say phosphate, I am mainly concentrating about the phosphorus element because oxygens will balance. So that is why I'm saying only mainly phosphorus. Also for ATP ADP relations. Okay, we'll sum this up once again. There is glucose, there is fructose. Both enter glucose 6-phosphate, hexokinase is there, glucose 6-phosphate, and then fructose 1,6-phosphate, bisphosphate, triose phosphates. Two things are there. Either it can be an aldehyde with 3-phosphate or an acetone. That is, ketone. Dihydroxy acetone phosphate or glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate is also called phosphoglyceraldehyde. Phosphoglyceraldehyde PGAL, and this PGAL gets converted into bisphosphate Biphosphoglyceric acid. Here itself acid conversion has taken place. Note karo, note this bisphosphoglyceric acid. And after that, we remove the phosphorus to give us phosphoglyceric acid. And bisphosphoglyceric acid is 1,3,1,3. This is also important. 1,3,1,3. And then 3-phosphoglyceric acid. And then 2-phosphoglyceric acid. Phosphorus shift has taken place. Position isomerism. And after that, enolization, phosphoglycerate becomes phosphoenol pyruvate with water elimination and ATP utilization, giving us pyruvic acid. ATP utilization, I mean ATP synthesis, very sorry, ATP synthesis. And how are we synthesizing is ATP, removal of phosphorus, and we have got pyruvic acid. What has happened in this process? The 2 ATP was given and 4 ATP, we have taken 4 ATP because everywhere if you see there are 2 triose phosphate, each gives ATP, 4 ATP. 2 phosphoglycerate, 2 of them are there, so we, I mean 2 phosphoenol pyruvate, 2 are there, so we get 2 ATP. And we have 2 NADH. If you remember, if you see, observe this one thing, in respiration, we say we only care about NADH. Whereas in photosynthesis, it is you have to photosynthesis P for photosynthesis. So NADP will be the assimilatory power there. Please don't confuse this. Yes. So pyruvic acid has been formed. Everything is formed. Everything happy. Now we are going to see about anaerobic what happens in anaerobic case. Pyruvic acid is the key product of glycolysis. What is the metabolic fate of pyruvate? This depends on cellular need. There are 3 major ways. How many major ways? 3 major ways. Very important. They will ask directly how much major ways are there. These are lactic acid fermentation, alcoholic fermentation, and aerobic respiration. Nothing else. Fermentation takes place under anaerobic conditions in many prokaryotes and unicellular eukaryotes. You have to know all these both points also can be asked. You can do it out by logic, but it is always good to have this known so that you will be saving time in biology. Prokaryotes and unicellular eukaryotes. For the complete co-oxidation, you need organisms adopt something called the Krebs cycle, which is also called as which is this whole thing is called as aerobic respiration. Why? It needs oxygen. Fermentation. In fermentation, say by yeast, incomplete oxidation is achieved under anaerobic conditions by set of reactions where pyruvic acid is converted into carbon dioxide and ethanol. So yeast majorly we are getting alcoholic stuff. Pyruvic acid decarboxylase. Why are we having a decarboxylate? Ethanol, how many carbons? 2 carbons. Pyruvic acid, 3 carbons. You need to remove a carbon, so we're having pyruvic acid decarboxylase and alcohol dehydrogenase. We are having an acid group. We need an OH group. We need to dehydrogenase that, right? Alcohol dehydrogenase catalyzes these reactions. We have to change the group. That's why we have this alcohol dehydrogenase. Other organisms like bacteria produce lactic acid. Bacteria produces lactic acid from pyruvic acid. So the eukaryotic example, what is the eukaryotic example? Yeast produces ethanol. The prokaryotic example, bacteria produces lactic acid. Humans also produce lactic acids. Muscles during exercises, it produces lactic acid. What happens? Oxygen is inadequate, so pyruvic acid is reduced to lactic acid by lactate dehydrogenase. The reducing agent is NADH, which is reoxidized to NAD plus in both the processes. In both lactic acids and alcohol fermentation, not much energy is released, less than 7%. This is also very important. Very important. How much percent? 7% of the energy in glucose. Not not as compared to aerobic. 7% of energy in glucose is released, and not all of it is trapped as high energy bonds. Not all of it is trapped as high energy bonds. This is also very, very important. 7% you are not getting it as in the form of ATP, 7% is released. But in that, how much we are getting it as ATP? We'll see later. Also, these processes are hazardous. Either acid or alcohol is produced. Both of them are toxic, not preferred to be accumulated. So, what is the net ATP that is synthesized? Only 2. Why? The NADH which was synthesized again got back into NAD plus. We'll see why. You may think this is a stupid process. Why are we doing this? This is a stupid process. We'll see why it is so. What is the net ATP? Yeah, we have seen When one molecule of glucose is fermented to alcohol or lactic acid, yeah, that's what was the question, right? Yeast poison themselves when they have a concentrate alcohol concentration of 13%. So the percent cases, what are the percent problems we are seeing? Percent statements, 7% energy, 13% poisoning. These are important. What then should be the concentration, maximum concentration in alcohol is if they're naturally fermented? Obviously, 13% cannot be more than 30% because yeast will kill itself and it cannot give it in more concentration. But how are more higher concentrations produced? Very simple. By manual concentration. How are you manually concentrating? You can either evaporate water and concentrate. We'll see that all that in 12th standard. How do you think alcoholic beverages are? Okay, that's what I should answer. Sorry. What then is the process by which organisms can carry out complete oxidation of glucose and extract the energy stored in this stored to synthesize a large number of ATP? That is very simple. That is aerobic respiration. That is what we're going to see. It requires oxygen. Is the presence of that leads to a complete oxidation and release of carbon dioxide, water, and a large amount of energy present in the substrate. This type of respiration is most common in higher organisms. We'll look at in the next session. Before we go to the next session, we have to see the graphs. Graphs are like this hidden treasures. If you don't unlock the hidden treasures, you will mess up in your meat because they'll ask something from this and then we will be blank. They will put boxes like this and they'll ask what is A. So glucose, after double arrow, what are they showing? They are showing glyceraldehyde 3-phosphate. What is glyceraldehyde 3-phosphate? PGAL. And then they are showing 3-phosphoglyceric acid. What has happened here? Phosphoglyceric acid, if you remember what happened, phosphoglyceric acid has, what did it become? Bisphosphoglyceric acid. And bisphosphoglyceric acid becomes phosphoglyceric acid. And after that, there's an isomerization and then phosphoenol pyruvic acid, phosphoenol pyruvate, and after that pyruvic acid. And here they have only mentioned about 3-phosphoglyceric acid, PGAL, and PEP. After that, for pyruvic acid converts into lactic acid. How is it converted? By removal of, by addition of hydrogen. You are reducing it to form lactic acid. Here you're decarboxylating it and you're reducing it to form ethanol. So why are we actually doing this now? You need NAD plus. You need this. You need this compound. Where are you needing this compound? This is so messy, man. You need this compound for this conversion, this step conversion. This step conversion, you need this compound. And how would you be able to regenerate this compound? That is why all the energy is wasted. We need to continue this process. We need to keep this running. So that is why we are wasting the energy, wasting the assimilatory power. We are using this like this. I hope you understood this clearly. We are now moving to aerobic respiration. So before we move to aerobic respiration, we'll see how much are there. So we have completed around 40% of the lesson, I will say. 40% is completed. Aerobic respiration first, there is on this linking cycle, linking reaction thing, and then we have the tricarboxylic acid cycle, very simple. Electron transport chain, the most interesting part of this chapter. People get scared about this for no reason at all, very simple. And after that, there is no lesson at all. Like it is all very simple questions. Like you are doing yoga, it will be very simple. Respiratory balance sheet, very simple. Amphibolic pathway is just like an interesting commentary. Respiratory quotient is also very simple. So we have to, as soon as we cross ETS, there is nothing in this chapter. There is only in how many pages we will be crossing ETS? 1, 2, and in 3 pages we will be crossing ETS.